And I know technically.
Yes.
Do you mind if I just make sure the presentation is the correct presentation? That ease in my mind.
Consistently.
Okay, yeah, making sure. That it's still in there. Perfect. Cool. It looks good. All right, cool, perfect.
Is this a video?
Yes.
Okay.
We like don't need sound for it, but it'll just like play. Yeah,
Yeah.
it's like a music
hear it.
thing. But like, the music, I'm probably kind of gonna like talk over the video.
Okay.
If that makes sense.
Yeah, totally.
There's like 2 videos that I kind of just talk over instead of like needing sound for.
I always forget. Right.
I wish I could help because I also don't remember how to do it.
Uh-oh. There we go. So as you can kind of see from my trajectory in my career, I guess, so far, I've gone from identifying problems to implementing solutions. And that's kind of the route I want to take you guys through today. So let's start with the problems. Since 1990, we've seen significant collapses in our shellfish and finfish populations here in the Indian River Lagoon. Specifically, shellfish has decreased by pounds harvested by 72%, and finfish pounds harvested has decreased by 54%. So these decreases are of course attributed to overfishing, habitat loss, and degraded water quality. Overfishing, of course, we love our shellfish. We love eating the oysters, the clams, and we love catching random fish, different fish, taking pictures with them, whatever the case may be, commercial or recreational. A lot of people don't know this, but the small-tooth sawfish, one of the most endangered alive-ranked species in the Indian River Lagoon, or in Florida in general, used to patrol these waters here in the Indian River Lagoon on a day-to-day basis. They used to be everywhere. Now, if somebody sees it, you probably find it on the news. And last but not least, the water quality issues. As you can see from this camera image of a living dock, you can barely see the dock from about 2 feet away. This is due to the turbidity, physical particles flowing through the water, causing that cloudy effect.
Another issue that the lagoon faces is muck. What is muck? Muck is this fine grain anoxic sediment that's on the seafloor all throughout the Indian River Lagoon. This has happened over years of runoff from nutrients of like fertilizers and also decaying of algae and like grass clippings getting into our waterways. And so over time, it becomes this muck looking gross substance. I don't know if you guys ever been in the lagoon, but I've been in muck like up to my shoulders and not shoulders, up to my hips and everything, which is crazy. But you can see over here in this visual that back before 1950, the sediment on the Indian River Lagoon seafloor was kind of more of like a sandy sediment rather than this mucky stuff. And then in 1950, when urbanization in the area kind of grew, we saw more of this muck from all of that pollution and all of that nutrient runoff from fertilizers and other things that occurred after the population grew here. And from all of these issues, we created the Living Docks program in 2013, which is a citizen science oyster restoration project where we want to promote benthic growth to mitigate the water quality issues. And to achieve this, we have aquaculture grade plastic mesh mats that we attach recycled oyster shells to with UV resistant zip ties. We get these oyster shells from a shuck and share program from the Brevard Zoo. They go to local restaurants, get like oyster shells from people like eating the oysters and everything like that. And then they dry them out and they give them to us. And then we drill holes in these shells and attach them to the mats. The reason we use these oyster shells is because it's made up of calcium carbonate, which is a chemical composition, and it attracts other oysters and also other shelled organisms. So it promotes a bunch of different things. You can kind of see down here, all that stuff that it promotes. Kind of looks gross, but it's actually really important. There's biofilm, there's barnacles, and all that other cool stuff. And we'll talk a little bit more about those organisms later. And here's just a cool video of kind of how we create a living dock. Just press it once, maybe. Start the video. Perfect. So like I said, we get these oyster shells from the Shuck and Share program in Brevard Zoo. And we first start out by preparing the mats. So we get these oyster shells. This is like what they come in, big trash can full of oyster shells. They're very heavy. And then we take them over to our drill press and we drill a hole up at the top of these oyster shells. And this is how we put the zip ties in it and affix it to the oyster mats. So local community members have helped us out a lot with this, and also we kind of just go and bring our undergrad help from Florida Tech to kind of help get this process done, and then we usually bring it to the community centers like here. You'll see later that we have some already drilled oyster shells. And then this is a great example of how we attach the oyster shells to the mats. And then this is a good example of about 60 to 70 recycled oyster shells on a plastic mesh mat. And after we create this, we want to get the community involved, like I was saying. So we bring them to, this is just examples of just at different community events of, this is Melbourne Beach Pier with a bunch of mats where we're going to go and deploy them. And this is also a mat making workshop over at Florida Tech and shell drilling, a little more examples of that. And then we deploy them. In this video, it's going to be an example of Aquarina, which is a local community in Melbourne Beach. And they had about 100 to I think like 130 dock pilings. And we deployed this, I believe, in 2019, 2020 time, and then also in like 2022 again. There's a lot of dock pilings. We can do it all in one day. So we have local community members from Aquarina help us place these mats out on the docks. And then some of us get into the water and then we affix them onto these pilings with UV resistant zip ties, and they pretty much stay. We'll talk about a little bit more about how well this project's doing in a couple more slides. And then after we do this, we also do a quick little assessment after a couple months. We usually go out just to make sure they're still doing well, that no storms have like disrupted them, and how much growth and like kind of what stuff is growing on them. This is a good example of kind of the poor water quality we were talking about earlier. Can't really see anything until you get like inches away from the oyster mat. And then this is also after a couple months, you can see that green stuff, which is green algae, and then a bunch of barnacles and oyster growth on this mat. So this mat is actually doing pretty good, and we like to see that.
So some of the benefits of wrapping pilings are the fact that we're keeping them away from that mucky bottom. Muck is anoxic, so there's no oxygen in it whatsoever. When organisms try growing in it or surviving in it, say with our oyster restoration bags or crates, chances are they're going to suffocate because they have nothing to breathe. They are animals. They still need oxygen just like you and me. So attaching these mats to the piles themselves keeps it out of that muck. Another benefit to the Living Docks program is that we don't need permits. We're attaching these mats to already established docks. So everything that we're using is already permitted. There's no reason for anybody to be concerned. We already did load bearing, load testing. There's absolutely no damage to piles. So another benefit is that we encourage that healthy, diverse community of local organisms, which we're going to talk about here in a second. And last but not least, it's easy to assemble. Anyone in the community can come out, help us, any age, any time of life. They can come, help us out, enjoy a day, and really feel and see that they are helping the ecosystem. So obviously we're talking about oyster restoration, right? And the reason why we're so focused on oysters is because one single oyster is able to filter up to 50 gallons of water a day in optimal conditions. Now, that's just one oyster. Imagine 100, 1,000, million even. That water filtration capacity is going to skyrocket. But the most important thing to remember about the Living Docks project is we're not just focused on oyster restoration. Sure, it helps the lagoon a lot, but we're also focused on all of the other organisms that come with these communities. So you have our barnacles, our encrusting bryozoans, mussels, tube worms, sea squirts, anemones. All of these organisms are filter feeders, and they all pull different size particles at different filtration rates. So everything all together helps the lagoon in its entirety.
I wanted to show you guys kind of an example of a long-term assessment. So this is the mats. This is an example of one mat from the Aquarina location that was shown in that video earlier when we deployed them in 2019 or 2020. And we actually took the one mat up about a couple weeks ago on Earth Day. And we wanted to see like how well they were doing. Really haven't assessed them in a while. And you can see on the bottom here, these are huge, full-grown oysters. These are like the biggest oysters I've honestly ever seen. It was awesome. And so we were talking about earlier how much oysters can filter. They're kind of like the prime top of the pyramid filtration organism compared to all the other ones. So you can see that there's just on this mat, probably 10 to 15 fully grown oysters that you can only imagine how much the impact of 100 mats have on that whole area. Especially in that area, it was a pretty mucky bottom. Like I said, in that area, I was up to like probably my waist in muck. So I feel like this is really important and helps that area the most. And not even just the oysters, there's also a bunch of different growth on these oyster shells. See that orange stuff, and it's a mix of sponge and encrusting bryozoans. And there's also some barnacles growing on it, and also some colonial tunicates and stuff like that, which are all filter feeding organisms and all very important. So we also wanted to kind of assess like how these oysters were filtering the water. So what we did is we put recycled oyster shells out for 5 months, and we took them back in, and you can kind of see here like all the growth on it. Like down here is what we call like a sea squirt. I'm sure you guys have seen those before. Kind of look like little snot balls. And then there's a bunch of biofilm, red algae and stuff like that. And then we put them in tanks for,
Oh, no.
oh no, okay. There's a little video here, so I might just have to put them in tanks for a couple hours, and you'll see in the video in a second. Is the video not there? Oh no. Oh, there we go. Perfect. All right. There we go. Price is averted. All right, on the left will be a tank with no oysters in it, and then on the right will be a tank with those oysters that we saw on the previous slide. And you can kind of see there's a picture of a fun little manatee in the back, and on this side it's kind of blurry and turbid and kind of not fun looking water. And then over here on the right you can kind of see as the water gets clear from just 5 oyster shells. And we don't even have fully grown oysters on these yet because it's only been 5 months. You can kind of see the impact even only after 4 hours. It almost looks clear enough to drink.
So as you can see, we've done some experiments with the water filtration rates of organisms from our living dock mats. You can see in these bar graphs at the bottom, each one is from a different location. So the one on the far left is from the Banana River. The 2 on the right of that are from the Indian River. And then our control is just a regular oyster species by itself. You can see in every single one of these experiments, the amount of chlorophyll concentration decreased. Now, the implications of this are that our living dock mats, especially in higher quantities, might help mitigate or even slow harmful algal blooms. So the decrease in chlorophyll really tells us that we're doing something right. And as I mentioned earlier, the entire purpose of my thesis project is to evaluate how much of a habitat we're creating for mobile organisms around the lagoon. So my objective is to essentially use camera data rather than physically catching these animals. I want to see how they're behaving, what they're doing at the dock, and how they're using the dock for ecological or habitat benefits. Are they hiding? Are they feeding? Are they using it to graze, etc. Another thing I really want to look at is some of the invertebrate communities. I want to see if we're seeing any juvenile spiny lobster. Those are economically important here in the Indian River Lagoon. I want to see if there's any pipefish coming around. These are an endangered species that we have occurring in the area. And I want to see shrimp and crab, which are main food sources for a lot of different important fish species. And some of my preliminary data that I recorded last year, I did see a high percentage of sheep's head around our docks. I also saw snook and a bunch of anchovies, which are essential, yes, absolutely. So as you can see, we're already attracting a wide diversity of organisms to our docks. And with a little bit more data and more extensive data, I'll be able to produce some better graphics on what we're seeing down there. Maybe some better pictures, some videos.
And to go a little bit more into my thesis project, I looked at stainless steel non-plastic alternatives for living docks. The 2 stainless steel materials that I observed was the 316 stainless steel cable ties and also 316L 18 gauge stainless steel wire. And we deployed these at 4 different locations throughout the Indian River Lagoon. And we kind of wanted to test the durabilities of these materials to see if they can withstand different environmental conditions like salinity, dock protection. So we had docks that were protected by like land and mangroves. And then we also had unprotected dock locations, which were more open out into the lagoon. So it was more affected by the wind-driven wave actions that are out there. And so over time, we saw some failure with the stainless steel cable ties. So you can kind of see over here in that first little image, where the red circle is, we saw like the stainless steel material almost ripping through the mat, which kind of makes sense. It is a plastic mesh mat and the sides of the cable tie are a little bit sharp. And this happened only after a month in a protected location. So maybe a storm event kind of caused some high wave action in that area. And then over here on the right, you can kind of see just the stainless steel cable tie material left and no shell. So there was some sort of corrosion possible issue or also high wave action that kind of pulled this cable tie apart after they were connected. And so after all this, with a little bit of a cost analysis, the stainless steel cable ties were a lot more money. They are great for citizen science because they're kind of easy to use. It was easy for people to attach them to mats. But the 316L stainless steel 18 gauge wire was cheaper and also more effective than these marine magnets. So, I know it's okay. It's fine, we're good. We can go next. So like I said, we're trying to turn this into more of our citizen science, trying to find ways to put the 316L 18 gauge wire onto the living docs mats where you guys can attach. So we've got a bunch of different tools like wire twisting tools, seeing if gloves help because I've twisted them. After like 100, your hands get a little tired, get some stabs by the wire and stuff like that. So we're trying to figure out ways to kind of avoid that and to keep the method that we had all along, which was a simple method with little maintenance. And we want to use you guys and the citizens to help us make these and on the end, just improve the Indian River Lagoon's water quality. And yeah, so we've also partnered with a bunch of different community groups such as Girl Scouts and Boy Scouts. We just had an event with the Barrett Island Center, if you guys are familiar. We had a map workshop and a map deployment last month. We actually have one coming up on May 15th and May 22nd. If you guys are interested, let me or Dante know, and we can kind of tell you guys more about it. We've also done events like a day in the life in the Indian River Lagoon, where the grade school around Brevard County comes out, and we have a bunch of different stations. This is at the Melbourne Beach Pier, where they talk about water quality, the sediment within the lagoon. We get them in the water. A couple months ago, I got them in the water with me, showing them the mats, showing them what's growing on them, kind of teaching them what the issues are within the lagoon and how they can help fight this.
So these two maps here show kind of a good idea of how much the project has grown since 2020. Back then, there was only about 25 to 30 mats, 30 docks installed around the area in the Indian River Lagoon. As of 2025, and that is actually an outdated map, we have around 50 to 60, not including the ones that have been installed by Indian River Lagoon that I have in pink here. So just the green dots are the ones that we at Florida Tech have installed. But the Indian River Lagoon has partnered with us, or Indian River County has partnered with us, so they're taking over some of those responsibilities for installing in that county. This growth alone truly shows the effect that we are potentially going to have and are currently having now. Any restoration scientist or restoration person will tell you the larger your restoration project is, the more effective it's going to be.
Some recent and ongoing research we've done with the living docs, we have tried to do plastic mat alternatives in the past with cathodically protected steel, trying to make this a substrate for oyster growth. So it's kind of like bio-rock, you guys are familiar with that, which was very successful in promoting benthic growth, actually more successful than the plastic mats. But it's not a great citizen science alternative because it's heavy, sharp edges, and you kind of need a solar panel. It's like an impressed current kind of system. So it's a lot more complicated than the simple design that we were talking about earlier. But it is a cool idea and could definitely be more of a thing for not citizen science initiative, just for restoration efforts in general. Also, before my thesis project about focusing on stainless steel materials, we had another plastic zip tie alternative. So we looked at 316L 22 gauge wire, which is kind of where I got my stainless steel from. Jute, coconut wire, and bees, which is like a potato starch material, but in a form of a zip tie. These were all cool ideas, but after 18 months, we did see that the only material that was left on the mats was the 316L 22 gauge wire and the plastic zip ties, which we used as a control. So that's kind of where we went to the direction of what stainless steel would be the best for the citizen science initiative.
And as I kind of talked about earlier, I want to use camera data to evaluate the ecology of the mats of these areas. So to do that, obviously, I have to find a way to navigate through the turbidity. So I have developed a camera box that will allow me to see through that turbidity, cut through the turbidity by looking through a liquid media such as tap water or DI water. This is going to allow me to get closer to the dock in my camera video without kind of messing with their daily functions and how they behave. So of course, I'm looking at those ecological impacts and any biodiversity index that I can muster up. And I'm going to compare that to dead docks, docks without living mats. And as we discussed earlier, we did do those filtration rate studies, and we are planning to do some moving water filtration rate studies within the next coming years. We have submitted a doc or a grant application for it. We have no idea if we won yet, but if we did, then I'll be sticking around for my PhD for that. But other than that, these are some of the ongoing researches that we have going on. So without further ado, let's make some mats.
Let's make some mats. Yay.
Great job, you guys. So we have a ton of questions in the chat box. Does anybody have questions that I can come around and pick up? So, okay, I'm gonna ask a question to get us started while I am coming around and picking those up. So the first question that I am gonna ask here is historically there weren't oyster islands in the Indian River Lagoon, kind of like up in Flagler County near St. Augustine, but then where they were harvested and destroyed by industry. So I think this is asking is like, what did the historical population of oysters look like in the Indian River Lagoon, and how might that have varied from maybe other environments or specifically estuaries in the east coast of Florida?
So there are a whole lot of different research articles on shell middens. Shell middens are piles of shells from indigenous tribes back in the day. And these shell middens prove that oyster harvesting was a super common act, and it's how they survived back then. They also used some of these shells as tools. So the fact that they were harvesting in those high numbers, enough to make mounds and piles of these dried oyster shells, essentially fossilizing oyster shells, really indicates that the population of oysters in the Indian River Lagoon was extremely high back then. Of course, since then, industry has overfished the entire area, so we don't know exact extents, but we know that the entire Indian River Lagoon was coated in benthic and hard bottom. That's also shown in the muck that we discussed earlier, especially those cores, right? The sediment samples before 1950, the Industrial Revolution, really when everything started picking up. Before that, those sediments were white, clear, looking like sand, essentially, more so than the dark, black, anoxic muck that we see now.
I kind of have a follow-up question to that. Great response. But can you maybe explain the difference between what, like, if oysters in the lagoon were subtidal and intertidal, and what the differences between those two might look like, just for general audiences that might not be aware. Okay, great. So I can answer that question.
Okay.
So that was more of a broad question. Different populations of oysters throughout the Atlantic coast might be intertidal or they might be subtidal. So that means that either the oysters can be submerged in water for full time, or with the tides, they can only be submerged for part of the time. So in the Indian River Lagoon, I believe that oysters were submerged full time, whereas the question that somebody asked about in Flagler County with salt marsh, and we're thinking Spartina grasses like salt marsh, oysters in that community are going to be subtidal where they're, or excuse me, intertidal where they're exposed at some points to the air and then they're underneath the water for part of the time. So most of the oysters in the Indian River Lagoon are going to be fully covered with water. And the reason I wanted to ask that question is because with this program, they're putting the oysters on the docks here. And the person asked about oyster islands. It is unlikely that we had oyster islands in the lagoon because they are completely submerged in water. That's why I wanted to ask.
Thank you.
Thank you.
Sorry. Okay. Next we have, oh, this is related to what I just said. Is there a depth that is best for the mats?
Sure. So we like to keep the highest shells right at that high tide watermark, just so that we get the full community. We have our low tide communities, our submerged communities, and we have more of our splash zone communities, barnacles, things like that, that can withstand those dry conditions when the tides go out. I would say right in the middle of that tidal zone is where we try to put the mats. It's just It depends on when we install them, how deep we have to reach down, and if we can reach bottom. Sometimes these docks, we can't really stand. The water is high or the current's moving really fast. So we just have to kind of get the map where we can get it.
The only thing we really do is try to elevate it off the bottom of the floor, like the sea floor, because it's either mucky and also just competition with other benthic organisms, trying to give them their best shot that are the people that are living, the organisms that are living on the oyster mats.
I have two questions that are kind of related to each other, and I find these really interesting questions. Also related to the mats themselves. Do you have estimates for how long the mats will last? And then in addition to that, what ranges of salinity have the mats been tested in?
So we want these mats to kind of just almost sediment onto the dock structure, so we don't have to touch them over time. Like I said, this that Aquarina dock was from 2019, so over 6 years. So in the grand scheme, we kind of want them to last us forever, pretty much, as long as they can, just to almost form their own community. So that's kind of the main objective here. So it almost becomes one with the dock. So it stays as long as the dock's there, pretty much. And then the other question was asking, remind me? Salinity. Yes, we've actually tested them in quite a wide range of salinities. My project in general, we tested in kind of where Crane Creek, I don't know if you guys are familiar with that, over by downtown, Crane Creek meets the Indian River Lagoon. We have a Florida Tech Merton Center over there, which is just kind of our marine station. And the salinity, like the surface salinity there ranges from like, depending on how much rainfall we have, between like 3 to like 15. So it's pretty low salinity levels. And we still see like tube worms, barnacles. I still see oysters there. We still see kind of similar organisms, but organisms are salinity based. So we see more like tunicates and other things like that, more in higher salinity waters. So we've also tested in up to like, I've seen in my project, we tested up to 27 salinities, PSU, parts per thousand. And we saw a bunch of little different organisms. So we saw like different sea squirts and kind of more abundance of maybe barnacles and other stuff like that. So it's almost like the organisms are more affected by the salinity, but the mats really aren't, especially with the plastic, they really aren't. The salinity might more affect the steel that we were talking about because it can cause accelerated corrosion over time.
That was an excellent response. And one thing that I do want to note here is that I would like to remind everyone, both online and in person, hopefully we're all aware, the Indian River Lagoon is an estuary, which means there's both freshwater input and saltwater input. And so there's going to be a range of salinities within the estuary, and having that range of salinities does constitute a healthy estuary. In the Indian River Lagoon, of course, we have mangrove trees, we have oysters, and a variety of other life is in addition to all of those things, and and seagrass as well. And those organisms have a specific salinity that they prefer over others. So depending on those micro changes in salinity between, maybe 27 at this part of the lagoon, but down to 20 at this part of the lagoon, there might be organisms that prefer those different areas. So this entire range of salinities is normal at different parts of the lagoon. So I just would like to remind everyone that.
Yeah, and also like in Melbourne area, it's kind of more far away from inlets. So I've seen like Melbourne Beach Pier, which is open out into the lagoon, doesn't really have a freshwater input like right there. It's kind of more medium salinities, but like I have a location down by Sebastian Inlet, and those salinities are usually higher. Like every time I went, like every month, they were usually around like 27 and stuff like that.
Yes. And so one thing that in this salinity question leads into the next question. And they want to know specifically, how does freshwater potentially impact the intrusion of freshwater into the lagoon? Say too much freshwater from, releases of Lake Okeechobee, or too much freshwater from other potential sewage plant overflows, or any number of other things, rain, storms. How can freshwater impact the organisms that live in the lagoon?
Yeah. Do you want me to go?
Most of the benthic communities that we talked about here, the filter feeders, most of them are going to be high salinity based. So most of the freshwater filter feeding communities are going to be microbes or biofilm based. So with high doses of freshwater input, on a consistent basis, a lot of the organisms that we see and quantify aren't going to be able to survive in these conditions long term. Fortunately, a lot of these fluxes of fresh water, they're not permanent, and the salinity does come back most areas. So that's also a reason we don't go up into tributaries and start applying these dogs everywhere. They are selective. We have to do site inspections and stuff like that before we actually deploy.
And also the freshwater like outputs are kind of where like the nutrient runoff happens. And so it's like more of that and more pollution can lead into the lagoon and also into those areas that are kind of at the convolvence of like Crane Creek and the Indian River Lagoon.
Nice. Speaking of water quality changes, water quality in general, can you tell us about some of the monitoring that y'all might have done at the Aquafina site and what that looked like over time, including things like pH, nitrogen, phosphorus, any salinity, any of those markers that y'all might have looked at after the Living Docs was implemented?
We have not looked too much into like nutrient runoff and kind of the nutrient load after we put the operating docs in. The operating docs were a little bit before my time, so I'm not sure like the levels of at that time. But now when we go out and do my projects, we kind of measure like the salinity and the turbidity. Turbidity is kind of the most important. We kind of a higher level of turbidity or higher number of turbidity kind of shows that there's more organic matter floating around, how it's not as good. We kind of see that more in like the freshwater areas. So like at our Crane Creek location, we can kind of see that the turbidity levels are a lot higher, but then we go out by the ones by Sebastian and then usually they're lower and you can actually see where you're walking, which is nice. Sometimes you can't always see. You're perfect.
Nice, nice. I do have a few more questions here specifically related to the plastics aspect of the project. So, plastics are a prevailing part of marine restoration, but there are also simultaneously concerns of what that plastic could be contributing to the environment. Do you see any tearing with the zip ties or the plastic mats themselves?
We have seen some tearing with the plastic, like when you attach it, like if there's a storm event, we have seen that occur before. But usually it's kind of like a big storm event, like a hurricane, like once that we get like once every 5 years or something, like a big event like that could happen if these mats haven't fully like cemented onto the pilings. But after about a year and a half, they should be good even through storm events. But it's more like recent deployments that could be affected by like wave energy and get ripped and torn kind of like that, if that's kind of what you guys were asking about. Yeah.
And then specifically related to some of the other alternative materials, say the steel, you said, did you say stainless steel?
Yes.
Yes. Can you quantify exactly how much better that might be than the plastic alternative?
I don't know if I can quantify because what we're using UV resistant zip ties and also aquaculture grade plastic mesh mats. So this kind of material doesn't necessarily break down, especially after they cement onto the piling. So it's more like an issue, more we like kind of produce more waste, especially with the zip ties, we kind of cut the ends. That's kind of more of the concern rather than it actually degrading in the marine environment. The stainless steel is also a great alternative. It's more like the citizen science initiative, that's the problem. And I mean, we don't want it, you also have less waste with the stainless steel wire. So what we did, we just got a big spool of wire and we cut it. So there's no waste there. So it's kind of, there's like good things and bad things about both materials.
Nice. Before we move on to our next question, does anybody in the audience have any more questions that they would like to give to me? Okay. So one of the last questions, not quite the last question, but one of the questions is, and to be totally honest, if y'all don't know the answer to this, I'm happy to defer to this question. But have y'all tested any of the benthic organisms that are settling on the mats if they contain microplastic particles?
Actually, we have. The person before me, Honor, I think she actually worked at MRC. She looked at the gut contents within sea squirts and she saw a lot of microplastics. I don't know everything about that project because she's not here anymore, but I do think she saw a lot more microplastics than to be expected, which is kind of sad. So in these organisms, there's definitely microplastics. You do have to think about it. When these organisms are filtering water, they're kind of catching all of like the gross stuff in them, so they're probably catching a lot of the microplastics, just like fish, just like us. It kind of runs down the chain, like we're gonna eat, ingest microplastics from the fish going through the water. So it's kind of just a cycle.
Yes, and that was a beautiful response. I'm gonna elaborate on that just a little bit. The question specifically asked about oysters, and I wanted to expand that a little bit more. Speaking of oysters though, and I know we all eat oysters, I spent 4 years studying microplastic particles in oysters and counting them, over 12,000 individual particles in 6,000 oysters. And so this is kind of an idea, right? Like there are almost always certainly going to be microplastic particles in filter feeding organisms because they are so ubiquitous in our marine environments. Now, all of that to say, how many of those can be attributed to the zip ties and the mats? Who's really to say? Can we tell? Maybe, kind of. Excuse me, but even if we could send those samples to a lab to determine what kind of plastic is being used in the zip ties and the mats and whatnot, that there's also already microplastic particles that exist that are going to be composed of the same material. So it is possible to study that, but it is something that like would be hard to do, would be difficult to do. And then the last question we have, which is I think a wonderful question. If there are individuals who would like to make their dock a living dock, not just in Brevard County, but throughout the eastern coast, or the lagoon itself, how might they be able to do that?
So the Living Docks program kind of does Brevard County and also Indian River County. If you are living in Indian River County, you kind of can reach out to Indian River County. You can just look up Living Docks. Indian River County, I think they have a website, person you can email. And then for us in Brevard County, we have some flyers here. I'm sure it might be a person online. Maybe we could share it also as well. If they have it up here. You can follow us on our Instagram or Facebook. You can DM us on there and reach out to us. We also have our emails that we can share, and you can reach out to us or one of our PIs.
Yes, and when I send the thank you email tomorrow, I will make sure to include their emails in the thank you email. So if you are interested in asking a friend to do a living dock, or you do a living dock yourself, their emails will be in there so that you can reach out to them and ask about how you might be able to implement this program. Before we move on, are there any more questions today? All right. Thank you folks so much. I very much appreciate y'all coming out and being a very engaged audience. Let's give a hand to our speakers. I am going to take this time to go ahead and shut the Zoom webinar off. If you would like to stay and build an oyster mat, please, please, please hang out. As always, feel free to mingle, grab some coffee. Bathrooms are in the back. And thank you again for joining us today.
Thank you guys. And we'll